Those of you who are streaming in from the larger Jefferson universe, you're in the right place. We're getting ready to start our presentation. This is the second installation of Innovation Engagement, our speaker series. We have a couple of students that are taking this for credit. Those of you that are taking it for credit and you're physically here, just please check in at the front desk so we can note that for the record. Today is kind of an extraordinary day because we have with us somebody who made the dream come true. And there are lots of people in this room and in the larger research universe that are working to build successful biotech companies. 20 years ago, it was still challenging to do, but it was much more doable. These days, it's much harder. It's much more resource intensive. The challenges associated with with getting people to believe in your vision are increasingly significant. The bar gets raised higher and higher. There are lots of stories that some of you can tell about your own experiences in trying to move your own research along the line of success and commercialization. But today we have somebody who's actually done it and done it in a pretty big way. Many of you have been reading about Spark and Jeff Marrazzo, who is the CEO of Spark. Spark emanated out in the other corner of the science universe out of Children's Hospital in Wistar. They, right? Do I have that right? Endpoint. In full disclosure, there's always a Penn issue here, but many of us are Penn grads. So Penn, Wistar, and CHOP, and so it speaks to the kind of interconnected collaboration that really makes fast advancement possible. So today we're going to hear from Jeff Marrazzo. They very recently came up with a pretty major discovery that's moving into commercialization or is ready to be used in patient circumstance. It's a treatment for a unique form of blindness. So when you talk about a game-changing biotech, you're looking at a guy that's running one. So without further ado, Jeff Marrazzo.
Thank you. It's great to be here. Thanks for the invitation. It's been, and I'm going to share a lot about the ride and the journey with you today. But it's amazing to sit here today and stand before you and talk a little bit about where we were 5 years ago and where we are today. And part of what which is why I'm excited to be here. Part of what I believe and have believed since we started the company, and I'll touch on this as we go along, is that this city in particular has all the elements of the ingredients to build multiple versions of companies like Spark. And part of what our mission was to actually build it successfully here in the middle of the center of Philadelphia. We're in West Philadelphia specifically. But to show that you can actually be as successful, if not more successful, than some of my colleagues who operate such businesses in other cities. And I think part of the reason that that's possible here in Philadelphia, and one of the most important elements of that, is all of you sitting around this table and institutions like this, of which there are so many of them in this city. So I will touch on that, but that's in particular why I appreciated the invitation, was excited to be here. So I'm going to spend about 20 or 30 minutes telling you a little bit about the story of Spark. I will probably actually do it in a bit of a reverse order, where I'm going to tell you about what the company is up to today and what we've accomplished, share with you, since a lot of you probably have an appreciation for not only the science and the medicine. I'll go into a little bit more detail about some of that in telling you about what we've accomplished, and then I'll spend a few minutes talking about the journey towards the end of it. But I wanted to give you a sense of not the destination, but the waypoint that we're at along this destination and our vision. So you'll hear me talk about this a couple of times today, but the overall goal of our company, the vision of our company, is a world where no life is limited by genetic disease. And we are aspiring to do that with what I believe is certainly a definable, transformative technology in gene therapy technology. And I'll talk more about how we got started and what that entails and show you some good examples of it. But ultimately, for everything we do, it comes back to that vision and the patients who are affected by genetic diseases throughout the world, and how we can ultimately change the course of what they choose and how they choose to go about their lives, and create a world where no life is limited by that. So because we're a publicly traded company, I'm required to tell you that you should read our formal prospectus documents if you're interested in trading in our stock, and there's a whole bunch of risks that you'll see when reading that. So I'm going to begin by just framing what actually rare genetic diseases are and the consequences of them in aggregate. And it turns out there are about 30 million people in the United States that are affected by a rare disease. And that's actually, as we all know, about 1/10 of the United States population. 80% of those diseases have a genetic underpinning, and 50% of the people affected by a rare disease are children. So not only do you have the large majority of rare diseases having a genetic cause, but it obviously disproportionately affects people who are at the developing stages of life. And obviously the consequence of that in terms of someone's life is even more profound in that instance. Where am I pointing this? Where's the best? At the computer? Okay. So as I said, you have about 7,000 rare diseases, and today about 400 of those have an existing treatment. So actually the large majority, the vast majority of them have no treatment. And as I said, 80% of those have a genetic origin to them. And then when you look at what has been attempted within the field of genetic research and gene therapy in particular, we're about 40 to 50 years out from when this idea of saying, hey, if we could actually map what it is that is the underlying genetic cause of a genetic disease by understanding the DNA that was causative of that, could we actually use that as a way to treat a disorder, actually have that be the active way in which we treated the disease? So about 40 to 50 years into the research of this, there have been 2,300 trials that have been conducted, which is an amazing amount of trials that have been conducted or are underway or are planned. And I'm proud to say, through the work at Spark and many people that preceded our work at Spark, the first approved gene therapy was for a genetic disease was actually approved in December of this last year, and it was Spark's gene therapy for a rare form of blindness. So you realize just how much work, how many people have contributed to getting this technology to this point in time, and just frankly how special it is for me and for us at Spark to be a part of the first one that has gotten to this point in time, and what that means for what we can do beyond that. And I'm going to talk more about that today. Just to give you some sense of exactly what our approach specifically is to gene therapy, we use what are called adeno-associated viral vectors. And so the concept for many years in this area of research, as we started to actually understand the human genome, whether that was within the last 15, 20 years, or could isolate individual genes, the question wasn't the knowledge that we had then about the genetics that were causing the disease, but how do you actually put that in or create that, take that information, manufacture the active agent, but what do you actually put it inside? And so the delivery question, what technology do you use to actually deliver it, has been one of the major challenges for this field. And people have tried both viral vector and non-viral vector delivery strategies. The technology has largely coalesced over the course of the last 10 years on viral vector strategies or viral strategies, with the two primary viruses that have been used most successfully, AAV and lentiviral vectors. And they have dramatic differences in pros and cons, and frankly, areas of strength in terms of applicability. One of the properties of AAV that makes it so attractive is that it's not pathogenic. It is generally associated with the common cold, but it has a high degree of tropism for a lot of different cell types within the body. It does, as far as all the knowledge that has been accumulated, and there's been more than 100 trials in humans done with AAV technology, and so far has been shown to be safe and had good safety profile. AAV has shown that it tends to, when it's dropped its DNA into a cell, that DNA that's dropped in exists extrachromosomally, so it does not integrate into the host cell DNA, and therefore when the cells split and divide, you don't actually maintain the active agent when that dividing process happens. And so we actually therefore target cells that are post-mitotic, long-lived cell types. So the retina, the central nervous system, the liver as one that doesn't turn over as much, at least is general belief. And so that's where we began life, and I'll come back to this in a few minutes. But AAV is the technology that we use, and we put inside of it, in all the clinical applications that I'll talk to you about today, a normal functioning copy or normal correct copy of the gene in place of the viral DNA. So I spoke a few minutes before about our vision, and you see that at the bottom here, our mission as a company, and the way we went out to build the organization 5 years ago was to create a fully integrated business that had the capability of doing discovery, of doing clinical development, but of also ultimately delivering these types of products to patients. And this is an incredibly exciting moment because since December 19th of 2017, just a little bit more than a month ago, we can now say that third aspect, the delivery aspect, is now real. We are in the process of finalizing the operational components of launching Luxturna, which is the trade name of the United States for our product for this rare form of blindness. But now this concept of not just discovering and developing, but delivering a medicine, not an investigational agent, but a medicine ultimately to people based on the power of the human genome and the information this is the moment that's first happening. And it starts to open up your eyes to what are the series of other things that we can do with the technology and the other ways we can deliver this. And I'll talk a lot more about delivery as well as what other areas we're working on in terms of discovery. And so we think about the mission of the organization on those three pieces: discovery, development, and delivery. And we think what we are doing are developing these types of treatments and delivering them in ways that are unimaginable until now. And when we think about that, it's not just about the science. It is very much about the science, but it's also about innovating in the way we do development. And I'll give you some examples in the way we worked with the FDA around our first approval, but also in the way that we deliver these products. And I'll talk more about that in the context of some of the announcements we've made recently around how we are proposing new ways for insurance companies to pay for these types of innovative one-time treatments. So the idea is, when we say treatments in ways unimaginable, we mean everything. We mean across the entire business. We want to keep innovating and thinking of new ways to do this, and this technology empowers you to do that. So this is a quick snapshot of what we in the biotech industry typically refer to as the product pipeline, and you'll see these things if you're familiar with biotech companies. What you'll see here is that, as I spoke about before, we have led over the last 5 years, we've let the technology and where AAV has the best applicability technically to work to define the three areas of the human body that we've focused on. And so, as I said, the retina, the central nervous system, and the liver are the three target organs or target tissues that we are actually trying to drive gene expression in by using AAV as a delivery vehicle to ultimately drop DNA into those cell types. And I'll talk more about each of these three in some detail. But you see they all have that property, as I said, of being post-mitotic, long-lived, and therefore enabling long-term outcomes to happen from a single dose. And I'll come back to that theme as we go through here. You'll see we have not only the approved product in Luxturna in the United States, but we have registration activities going on in Europe. And we actually just announced last week a partnership with Novartis for them to actually bring the product to patients with RP65-mediated retinal diseases, which is the disorder, outside the United States. And so we announced that last week, but we are still in the midst of prosecuting approval for that in Europe. And then we have 3 other programs in other inherited retinal diseases. We also have multiple projects going on where we are targeting the liver as essentially a depot to generate gene expression from, including hemophilia B and hemophilia A, and I'll talk more about that and show you some really exciting preliminary data there in terms of outcomes on patients with hemophilia. And then we have work going on in the central nervous system as well with 2 different types of neurodegenerative disease, one of them being CLN2 disease, which is a rare form of pediatric onset neurodegenerative condition known as Batten's disease, sometimes referred to that way. So that's a little bit about the portfolio, and as we get to the question and answer later, I'm certainly happy to answer any questions you have about that. But I wanted to give you a picture not only of what we have been able to accomplish with Luxturna, which is our now approved product for RP65-mediated inherited retinal diseases, but I also wanted to use Luxturna and the example as a way to give you a sense of not only how we've innovated across the business, but also just what this type of technology can mean generically, not just within Spark's hands, but more broadly. And so I think it's important, and this is Caroline and Cole, who were two people affected by mutations in RP65. But if you can imagine a world where there is the potential to deliver a one-time treatment, a one-time delivery of, in this case, 150 billion viral vector particles with a normal functional copy of RP65 inside it, imagine what that means not just to the life, first and foremost, of those patients and their family, but secondly, how does it change the way we think about medical practice? And third, how does it change the way we think about our healthcare system and how we pay ultimately for treatments? And that I'll get on towards that theme as we go along. Just to give you some context about the area of inherited retinal diseases, because it's probably something for those of you that are in your studies right now, you might have read a little bit about it. Sometimes this has been described historically as retinitis pigmentosa. That used to be the name that all of these were described. Over the course of the last 20 to 30 years, there has been a tremendous acceleration of our understanding of the genetic underpinning of various retinal disorders, and specifically these inherited types. In fact, the scientific community has found more than 250 genes that actually can cause degeneration of the retina in an inherited form, and it's estimated that there's about 100,000 people affected with inherited retinal diseases in the United States. Now I say estimated because and this is an important point that I'll touch on and come back to later, in general, people with these types of disorders are not diagnosed to the genetic specificity of their disorder, and that's largely because there has been nothing to do about it. There are no pharmacologic agents, or there were not until a month ago, no pharmacologic agents available to treat anyone with these inherited retinal diseases. And in the United States, as many of you probably are aware, our insurance system tends not to pay for genetic testing where there's nothing directly to do about it. And then on top of that, there's always the question between the patient and family and the physician about, do you want to know the underlying genetic cause of your retinal disorder and why you're going blind versus just that you have it? That world, I think, is starting to change and will start to change with the fact that there's now an approved product for one of these 250 different types of genetic diseases. And I think it will start to drive greater awareness and education about the importance of genetic diagnosis, and that that's the most precise and appropriate way to ultimately diagnose patients with these diseases. You see the severity of this and just how significant the impact is on the quality of life with that fact there at the bottom that it's actually a little bit hard for me to read from here, but that basically you see vision impairment or total loss by the age of 5 in nearly 3/4 of these patients with inherited retinal diseases. So this is a significant consequence on the life of these patients. So let me talk to you. I've mentioned RP65 a few times, and it is certainly a mouthful when you try to describe the indication for Luxturna, but it is for patients who have biallelic mutations in this gene called RP65. RP65 is both the name of the gene as well as the shorthand for the protein, which in this case is an enzyme. It stands for retinal pigment epithelium 65 kilodalton protein, so not a very creative name, but it's consistent with the molecular weight of the enzyme. And what this enzyme does is it plays a role in the visual cycle. And so many of you probably know that when light strikes the retina, there's a series of biochemical processes that ultimately turns that into information that is transported to the back of the brain or the occipital cortex, where you ultimately see. And so in that visual cycle, there is this layer called the retinal pigment epithelium that effectively acts almost as a set of nurse cells, because the photoreceptors, which is the layer stacked right in front of it, that area in green, is one of the most metabolically active parts of the body, and actually it needs help to process through a series of these different steps. And so what happens in people who have mutations in RP65 is that they don't have a sufficient production, or in some cases no production, of the RP65 enzyme, and therefore they don't clear a certain set of substrates that build up as precursors in front of that, and that cycle that you see gets disrupted. And so you have an immediate biochemical disruption, which causes a lack of the ability to perceive light and process light to the back of the brain where you see, but it also eventually leads to anatomical degeneration because of this substrate backlog, if you will, in front of that enzyme. And so the concept behind gene therapy was if you could take the information inherent in sequencing of this gene, which was we found out what the normal functional copy looked like, and deliver it to that space in the back of the retina and gets those cells in the RPE layer to essentially take up the normal copy of the gene and put it into its nucleus, again, extrachromosomally. Could you then use the cell machinery that knows how to produce the protein from that blueprint, in this case, the RPE65 gene, could you actually give that the correct blueprint and allow it to start to produce the enzyme? And that's obviously what we have strived to do, going back from the preclinical work to now the approved product. So just a bit more about the clinical presentation of this. So it actually has somewhat variable presentation. That's partially based on the fact that depending upon how much of the enzyme you are producing, again, there are some patients that have mutations that cause nothing to be produced, which we call null mutations, to some people that are producing a little bit. And depending upon that, you might have more well-preserved function for a longer period of time. But at the end of the day, in this disease, it progresses inevitably, depending upon, again, the severity. It might start sooner and go slower, or start sooner and go faster. But inevitably, you see progressive deterioration of vision and eventual blindness in these patients. And as I said previously, before December 19th of 2017, there was no approved treatment, not just for RPE65 types of retinal disorders, but any of these. I won't go through all this data because there's a fair amount to unpack here, but we conducted one of actually the largest natural history study of patients with this disease. It's a very rare disorder. We estimate that there's probably 1,000 to 2,000 cases in the United States of people with mutations in RPE65, and maybe 10 to 20 new cases a year. And so we did as large a natural history study that had been done, where we looked at the charts of these patients on a longitudinal basis over time. And you see that two things are happening. One, that they lose their visual acuity or their fine vision. In fact, I think on average, around 16 years of age, people are legally blind on average in this cohort of patients in the natural history. But then secondly, what you see happening even at an even more rapid rate is a reduction in the visual field, which obviously can impact your functional vision. Even if you have well-preserved acuity, seeing through a pinhole is not as functional of your vision as if you have a wide aperture of your field. And so you see those two things happening in parallel, and they're both part of what is a complex process of vision in general, and certainly in these patients. So let me give you a sense of what it actually means functionally. And this is a video, so let me just introduce it first. This is a video of a patient who participated in our phase 3 trial and was asked to navigate a course that was designed to mimic the activities of daily living in people who have this type of disease. And they're asked to navigate under different lighting conditions. My lavalier mic on now? You hear that? Okay. So I'll try to...
Start.
So what you'll see is this as a 6-year-old girl who is trying to navigate this course and is being asked to follow the arrows on the floor and step over the obstacles in front of her. So you'll see pretty quickly that not only is she not identifying the obstacles in front of her and trying to step and is not recognizing them and stepping into them, but she's also not following the arrows. Most importantly, what you'll also recognize is she's not really using her vision. She's really actually feeling her way through. And so I'm actually going to skip ahead and show you the same. This is basically the same patient in that same test, and then the same patient a year later after she received a single injection. So this is at the same lighting condition. The course is slightly different because they have very good kinesthetic memory. So we changed the course, but it's a similar difficulty. So you see now that same girl 12 months later, after a single injection, and it's a little delayed. But you see that not only is she following it, she's really using her vision. She's not feeling. So to go back and give you some sense of what that all meant, I skipped through some of these. What we actually used this endpoint. We developed it over the course of both the original work that was done in the academic setting through the work that we did at Spark. We actually used that endpoint as the primary endpoint in the trial, and the goal was to not only show that we were having an impact on the biology, but what did it mean? That's sort of so what question. And so what this course was designed to do was to show that under different lighting conditions, and specifically in darker lighting conditions, you could see improvements in these patients after a one-time injection. And what you see from this data, this was the original phase 3 readout of our efficacy. You see highly statistically significant p-values, both in their performance on the mobility test, as well as supporting 2 of the 3 supporting secondary endpoints, which captured aspects of the pathophysiology of this disease changing, as well as in some of those endpoints I mentioned before, like visual field. It was not statistically significant for visual acuity, and I'll come back and explain to you ultimately why that was. Ultimately, it's a function of the fact that this is largely a rod-mediated disorder, which largely affects the peripheral vision and your light sensitivity as opposed to your central vision. But we did see a trend in that data, and in fact saw about 8-letter improvements overall in those patients in that trend. So when you actually graph this out and look at the phase 3 results, and just to put this back in context, the video I just showed you was a girl trying to perform at 4 lux. On average, the patients came in and they couldn't pass the course at 50 lux. And then you see afterwards, on average, they're improving to more than 4 lux in terms of the average patient. And we saw statistically significant improvements. And in fact, the majority of the patients actually could max out on the test, which was 1 lux. And 1 lux is basically the light of like a moonless summer night, or if you're trying to navigate your apartment with a night light on in the middle of the night. So now people who initially had limited light perception and ability to perceive that light and how that converted into their functional vision, now in the case of the majority of these, they could actually perform at the darkest possible condition we used on the assay. So not only were we seeing a change compared to the natural history, which is again a slowly degenerative disease where patients got worse, we actually were seeing improvements compared to their baseline function when they came in. And this mapped well with what was called full field light sensitivity threshold testing. This is essentially a way to measure the retina's ability to perceive light. And whenever the medical people on our team often relates it to hearing. So in a hearing test, they'll ask you they'll turn down on a log scale the intensity of the noise, and you'll see when you can actually hear that. Do something similar on the retina, where you actually sort of blast it, if you will, with different intensities of light and see when that light is being picked up. And you can see, going back to the biology that I described before, that if you are actually improving that visual cycle by adding in a normal functional copy of the gene, you would actually have better ability to pick up that light. And so that's what we see. We see actually about a 2 log improvement on average in the patients in their ability to perceive light. This is a log scale in that FST test. And as I said, the visual fields in these patients, using one of the 4 measures that we looked at, which was the mean Goldman visual field, using what's called a 3,4E isopter, that actually doubled. And so patient who had the field, whatever they might have been when they started, we saw on average that it actually doubled in terms of the size. So when you start to think about, again, what do these individual ophthalmology endpoints mean, whether it's light sensitivity or field, the goal is to capture that all together in a functional endpoint, which is that mobility test that I described to you before. The safety data from the trial was also very good and encouraging. And obviously, given the nature of the technology of gene therapy, it was important to capture the safety data in multiple different contexts. There were ocular adverse events that did occur in patients that may have been related, although most of what we believe to be the adverse events were related to the actual surgical procedure to deliver the vector ultimately to the retina. And you see these written down here, and obviously they're important to understand when looking at the benefit of a treatment like this, what are the related risks, and that's what this page captures here. For the full information about this, now that this is an approved product, you should look at the full labeling information and not use the shorthand that I may have shared here today. So I shared with you this video before. Let me tell you about the second part of what's important about this, which is that those gains in function. So if you go back a couple more slides to this picture where you see that the gain is occurring, these patients are improving by more than 2 levels on that mobility test. We saw that gain be maintained, not only show up at around day 30 and appear again at day 90 and 180 and 1 year, which was the primary endpoint, but we have followed a group of 20 patients now out 3 years, and it has been maintained. And we have seen a cohort of 9 patients that were in a control crossover group out 2 years. And we continue to follow these patients, but the concept of being able to use the information from the Human Genome Project, put that into a viral vector, deliver that to a cell, and then let the cell machinery to continually produce the enzyme seems to be occurring because you have the ability to see these types of long-term outcomes from a single dose. Again, all these patients that I just talked about received a single dose into each eye, and they have not received another dose after that point, which is incredibly exciting when you think about what that means for, again, the patient, medical practice, as well as our healthcare system, and I'll touch on those aspects as we go forward. We think that Luxturna, because of that, represents a product that has 3 unique attributes that have never been seen before. First of all, we're showing gain of function, so we're actually improving function back in these patients. Secondly, we're seeing highly significance of the effect, and the effect is consistent. In fact, in our phase 3 study, 93% of patients showed a gain of function, so a high degree of consistency of effect. And then the durability of effect that I just talked about. And again, important to keep in mind, the durability of effect and the gain of functions all need to be included in the context of the fact that this is actually a naturally occurring, naturally degenerative disease. And so these patients, if they were not treated, would only get worse. And so when you think about these gains and the durability of the effect and that it's actually stable, all that is incredibly encouraging in the context of where these patients would have been otherwise. So what we have is we have data in earlier phase 1-2 cohorts, and we have information collected from a dose escalation study, but this is the phase 3 study, and that's the registrational study that we have followed patients on so far. So for example, just to give you context, at our advisory committee hearing in October, one of the physicians who had treated a boy about 9 years before in the phase 1-2 dose escalation study, where we didn't actually use the same endpoints, but he said when he sees that patient every year and even recently, his ability to function in a dark setting is the same today than it was 8 years before. But that's a physician speaking about his personal interaction with the patient. These endpoints weren't used until phase 3. So a bit more about Luxturna and its approval. So a couple of really important, notable things about this approval, not just the fact that it was the first gene therapy approved for a genetic disease in the United States, but we also were successful in being able to reach agreement with the FDA that the label should actually be described for patients who have mutations in a gene and not with a clinical classification. Because our argument was that this area of ophthalmology was moving away from clinical classification of disease to genetic classification of disease, not unlike the way many of you, if you're familiar with cancer, is moving away from organ system distinction to molecular subtype. We argued that you should actually classify this, and anyone based on the mode of action of this therapy that had mutations in RP65 should be applicable, whether they were called this clinical name or that clinical name or that clinical name. And that was important because when we did this natural history study, we actually found that in those about 100 people that were originally investigated for that natural history study, we found there was actually 27 different clinical names given to people who had mutations in RP65. And so that was one important advancement. We also were able to work towards an agreement with the FDA where patients as young as 12 months old could theoretically get access to the treatment. And that's the concept. The reason for that is that the globe takes some time to be developed in young children, and that the upper age, that there should not be an upper age limit, but rather the physician should have the ability to judge whether or not the patient had enough viable retinal cells to be able to be transduced with the vector. And so those were all important steps that we made, along with the description of the safety profile that should allow physicians and patients together to make their determination about whether they want to get access to this therapy. As I mentioned before, we're already in the middle of our registration process in Europe, and we expect to have an action in the third quarter of this year for Europe. And we just announced last week that we have struck a deal with Novartis, who has one of the largest, if not the largest, ophthalmology business in the world, to bring the product to patients outside the United States. Yes?
And a question, so for somebody who's say in their 50s who was legally blind because of RPE 65,
Yep.
could they then take Luxturna and gain sight?
Yeah, so we don't believe that there's an actual direct age relationship. And what the reason for that is every patient either has or may have different mutations on RPE65. And so the mutation leads to the stage of onset or age of onset and the severity of the disorder. And so you could take actually two 50-year-olds who have different mutations in RPE65, and they could actually have very different looking stages of their disorder. So ultimately it comes down to, and what the FDA was interested in, was giving the physician and these expert physicians who see patients with retinal degenerative disorders the ability to consult with the surgeon and say, hey, what do we think is the relative benefit-risk ratio? Again, the risks being largely surgical. What do we think about the risks of those surgery vis-a-vis the benefit of someone who has less cells left to get the vector, for the vector to transduce, versus someone that's maybe earlier in their disease and might have more cells left? And that's ultimately will be up to the physician, the physicians really, between the medical retina and surgical experts to determine. Just for fact and color, we had patients from 4 to 44 years of age in our trial. So it gives you a sense that it was obviously a fair amount of patients who were young, but there were people as old as 44. And that woman actually, who was 44, specifically wrote in to the FDA during our advisory committee about the consequence it meant to her. And so, that, again, everyone has a different, may have a different impact in that context. So I want to switch gears and talk a little bit about launching what is the first gene therapy for a genetic disease, and we're in the middle of literally doing this as we speak, so you're getting kind of live play-by-play on what this looks like. We have focused on 4 major areas to ultimately bring this to patients and enable our launch. The first is to drive the identification of patients. I mentioned before that of those estimated 100,000 people with inherited retinal diseases in the United States, a very small percentage of those have actually had a confirmatory genetic test. In fact, we actually did some mining of claims data, health insurance claims data, and found that maybe 5 to 10% of people who have retinal degenerative diseases have actually had a genetic test. And so that's not necessarily for RPE65, but more generally. And so what we have been doing is driving the education of physicians, trying to make sure that they're aware that there's now an approved therapy, the fact that this is a disease that could and probably should now be diagnosed at a genetic level, and to facilitate access to genetic testing. And in fact, we are offering a free genetic test for RPE65 so that patients can ultimately get access to that without a restriction from insurance coverage around the testing itself. So there's a lot of work going on in terms of driving and helping to have any patient out there that has RPE65 have the information that they have that, and then ultimately they need to decide with their physicians what they want to do. But we think it's important that people are armed with the information about their specific genetic diagnosis. The second thing that we're doing is focusing on treating appropriate patients, and that goes back to one of the questions that you just asked. Ultimately, that's up to the physician, but we are interested in arming with the physicians with all the information about the clinical value, both the benefits and risks of Luxturna and the related procedure, and trying to get as many patients that are interested to be treated. Of course, both because we want to make sure that this product gets to all the patients that want it, so they need to be diagnosed and then have the chance to get the information. And of course, from our perspective, it's not only establishing that gene therapy can be a medicine, but also that it can be a business, right? And that's part of what the success of this strategically for us is to show that so that as we go and look at other diseases, we have a model and a pathway to move that forward. The third is around the patient experience. We're doing a number of unique things in this order. We actually are ultimately focusing on the distribution of this product in a very small number of centers around the country. Because this is a one-time treatment, you don't necessarily need to have this available. Certainly not, you can't, not only if you wanted to, you couldn't make it available at your retail pharmacy, but we don't also want to make it available even broadly. We want to actually have centers that have had specific surgical training to be able to deliver this, and who have experts in medical retina disorders, specifically retinal degenerative disorders, have the genetic testing capability. And so we're focusing in on, I think right now our website has 7 or 8 centers around the United States that we are working with towards setting up as places that this would be distributed to. And what we are also doing in parallel is we've built a patient services organization that ultimately can help patients and their families navigate all the details of going and seeing not only their physicians, but engaging with the insurance companies and organizations, and ultimately give them support on the back end for getting access to the therapy. And I'll talk more about that in a second. One of the things we are doing uniquely, because this is a small number of centers that we're delivering it to, delivering the product through, we are providing for commercially insured people free support, essentially support and free cost for travel and accommodation for a patient and their family to those sites. And so the idea is to try to eliminate, again, the risks and the barriers to getting access. And the fourth area is ultimately market access. And this is, of course, one of the most, probably the area that most people have been most interested in over the last many years as we've moved this program forward. But we are working to not only secure coverage with some of the largest, but also local, regional, and both commercial and government payers. But we also have introduced a number of novel payment models, and I'm going to spend a few minutes talking about this. This goes back to one of the things I said about the mission of this organization, which is that we are about not only discovering and developing, but delivering treatments in ways that are unimaginable. And we have tried, in the context of this launch of Luxturna, to think and spend a lot of time with insurance organizations and payers around the country to come up with new ways that we think these types of treatments can be paid for and distributed throughout this country in the United States. And 3 things that we have done. The first thing that we announced this month when we announced the price of Luxturna was the idea of creating an innovative contracting model. I think many of you are well aware that drugs that are administered by physicians ultimately are bought by that physician practice or by the hospital, and then that organization, whatever that is, turns around and seeks reimbursement from the insurer. That's called buy and bill. And our belief for a treatment that ultimately was going to be a high-priced treatment because we're capturing all the value in a single payment up front, our idea was, is there a way to actually offer an alternative? We can still use that buy and bill model, but is there a way where we could actually find an option for institutions that were going to be a part of this that didn't want to shell out the money to buy an expensive therapy and didn't want the risk of carrying that expensive therapy or getting covered? Could we actually sell that product directly to the payer and have the payer purchase it from us? And so we're working on this idea of this innovative contracting model where the payer would actually purchase Luxturna directly from us, and it starts to actually cut out a fair amount of what ends up being cost and risk that happens in the supply chain of these types of therapies. Most importantly, and the goal of this particular model was to ensure that patients get access. And so there are 3 things that an insurance company has to do to agree and work through this model with us. The first is that the patient, that they cover Luxturna consistent with the FDA label. They can't create restrictions over and beyond that, beyond their own volition. So they have to cover it consistent with the label. The second thing is that they have to process the benefits requests of those patients in an expedited form. So they can't take 6 months, 9 months. They got to process it under specific terms in a rapid turnaround time. And the third thing is that they are capping the patient out of pocket to in-network maximums. And so the idea is giving patients, ensuring that patients have access, that they get access quickly, and that we can actually ultimately cap the out-of-pocket portion for the patients. And then we, within the commercial insurance, patients covered under commercial insurance, we believe we have assistance programs that can cover all the costs related to that patient assistance for that particular patient. So the goal is, again, to get access for patients while also taking out cost and financial risk for both the payer and the treatment center. Not everyone will avail themselves of this, but it's an option that we think is important. Yeah, you have a question in the back? This is incredibly complicated stuff, so I'm happy to answer it.
Right. Are you willing as a company to consider spacing out the reimbursement over months or years from the payer?
Yeah. So the question was, are you willing to space out your reimbursement from the payer over months or years? So that's getting to the third one on this list. The second thing on this list is what we are offering is to commercial payers today is what we're calling outcomes-based rebates. So the idea is that we want to stand behind the product. We believe that it works. As I told you before, 93% of patients in the phase 3 trial showed gains of function where it was working. So we want to stand behind not only the initial effect that it's going to work, but two, that it will continue to work over multiple years. And so we actually are setting up an outcomes-based arrangement where we get paid up front, but we offer rebates back if it doesn't work at 30 to 90 days, or if it's still not, if it hasn't maintained its effect at 30 months. So we're standing behind it through the 30-month cycle, which is roughly around the churn, if you will, for an insurer. The third thing that we're doing here that we announced in January is that we're under discussions right now with the Center for Medicare and Medicaid Services and are discussing a way that we can actually relax two of the current government price reporting regulations called Medicaid best price and average manufacturer price. And the goal of those two relieving of those regulations is to basically offer plans the opportunity to pay us over time. And so rather than have it be all paid up front, to have it paid over multiple periods where some of it would be paid up front, some of it would be paid upon initial follow-up, where we would again check whether the effect was there and we wouldn't get paid if it wasn't, and then get paid another period out multiple years where again it has to be maintaining its effect. And so the goal is again to stand behind the product and to create some relief of what payers called cost density, essentially having it all come in one period. And so those are the three things that we are doing, and they are three things that I dare say I don't think anyone has ever done before, and are really ultimately about trying to stand behind the product because we believe not only in it, but we believe in this technology to create long-term outcomes. And let me say a few words about that. My general view is that our healthcare system is set up for 20th century innovation. And my view is to build and push us to a healthcare system and a set of policies that are consistent with where our science and medicine is actually going in the 21st century. And if I could summarize the way that, my view, the way it works today, we largely pay on volume, not on long-term outcomes. And so my sort of whole pitch on all of this has been to at least get the deck stacked in at least even for one-time potentially curative therapies, at least have it even. I would dare say that we should stack it in favor of those over therapies that can't get and are not innovative enough to be one time, because I think that ultimately is what, if you ask and walk down the street to a patient, I'll tell you in a few minutes about this in the context of hemophilia, which I think is the best way to see it. If you ask a patient, would you rather have a one-time treatment, or would you rather take this therapy chronically for the rest of your life? We don't have to do much market research to figure out which is going to be preferable for the patient. So let me talk to you a bit more about that in the context of hemophilia, because I think it leads well to this, and it'll help me get towards the, yes, go ahead. Yep. So what we announced was a wholesale acquisition cost of $425,000 per vial, per eye, correct? Yep. And then when it's obviously factoring in the rebates and so on and so forth, but that's the whack. What was that? Say, repeat the question. What's the volume? It's 0.3 milliliters, or 300 microliters. It's 1.5 10 to the 11th. So it's 150 billion viral vectors injected in a 300 microliter solution. Yeah. So going on to hemophilia, and this will just give you some, yes, go ahead. Yeah. It's not. It's not chromosomal. It's extra. It exists extrachromosomally, so it doesn't integrate. But because you go into cells like essentially the retina is an extension of the brain, and as you know, your cells in your brain don't divide and turn over as far as we understand. Right? This is what your mother told you about not going out and drinking at night, right? So the idea is that you don't have turnover.
If everyone could please just hold their questions so he could complete his presentation. There will be time for
I'll go
questions at
quick.
the end. Okay, thanks. I
Yeah.
appreciate it.
So the concept behind developing a gene therapy for hemophilia, is rather simple, and the idea is that instead of having the standard peaks and troughs of giving chronic infusions of factor, which are essentially protein replacement, and you have to give them on a regular basis because there's a half-life of that protein when you inject it into the circulation. The goal is, can you actually give a single dose treatment which creates a certain amount of protein expression from, in this case, the liver, which is what we're targeting, into the circulation, and therefore have a line that's consistently active or a constant level of factor compared to the peaks and troughs of that. And you can see how not only does that create the potential to provide better outcomes, because you aren't subject to the risks of the peaks during the periods of the peaks and troughs, but obviously from a patient perspective, one-time treatment versus 70 treatments a year for the rest of your life, and then from the healthcare system perspective, one-time treatment versus paying, in the case I'll give you an example here in a second, about $406,000 per year was what was spent per year for the rest of their life, was what was spent on the first 10 patients in our hemophilia B trial. So we went back and looked at their charts historically. They were buying, they needed enough international units of factor to the cost of about $406,000 per annum. And so let me skip and show you what that meant in the case of this. So on the right side, what these patients, even despite having regular factor infusions, all the green bars on the right there show you how many bleeds these patients still had. So even with a treatment, and in some cases these patients are taking it prophylactically, in some cases they're taking it on demand, but even with that, that's the number of bleeds that they had. We saw a 97% reduction in the amount of bleeds. Again, all these patients got a single dose, in that case, 5 times 10 to the 11th vector genomes per kilogram of what they got. But post, you see one patient out of the 10, or actually this is now 11, one patient out of the 11 had a couple of bleeds, suspected in that case, is my understanding. And then the part on the right shows you that these patients on average, I think the average is somewhere around 70 or 80 infusions per year. So they're taking 70 or 80 infusions per year, they're still bleeding. The 70, 80 infusions per year are costing about $400,000. We saw a 99% reduction in the infusion rate. So effectively, 10 of those 11 patients have not, for the follow-up, which were about 13 years total of follow-up in these patients, we've seen essentially no need for factor infusions since that time. So just to give you a sense, we're earlier in our work in hemophilia A, but so far in the first 4 patients, tracking very similarly. And hemophilia A, you may know, is about 80% of the patients affected with hemophilia are affected by hemophilia A. We actually partnered the rights globally to our hemophilia B program, which is about 20% of the market, kept the rights to our hemophilia A program, which is about 80% of the market, which was a specific strategic decision we made. So let me just conclude by saying a few words about the journey, now that you have a little bit of a sense of what the company does, hopefully a little more than a little. The first 5 years, so we actually started the company on March 13th, 2013. So we're coming up on 5 years since founding the company. When we started the company, we had one program in clinical development. Today we have the first ever product approved of a gene therapy for a genetic disease. We have 3 additional programs beyond that in the clinic, and we have multiple programs in preclinical research across all 3 of those areas. We started in 2013 with $10 million that we raised. Since that time, we've raised another $848 million since that time. So we've certainly raised quite a bit of capital to fund this. This is a capital-intensive endeavor, as was discussed before. We've published 7 research papers, including in the Lancet, New England Journal, high-profile journals, and we have grown from 3 people in 2013 to more than 300 employees today. And importantly, have grown our footprint from what was, someone went and calculated this, 130 square feet of space inside CHOP to now about 100,000 square feet of space in West Philadelphia. So I'll conclude by just saying a few things about what I think has been a part of our success. And I think it starts with attention to detail. And for us, it's been about making sure that we pick and design these products and take care of every detail of that, not only the vector that we choose, what we put inside the vector, how we make it, how we formulate it. All those have been critical components. The second thing that's been important, and it was a strategic decision, even when we only had $10 million in the bank, was we decided we were going to go and build our own manufacturing facility, and we happened to have done it at 38th and Market in the middle of West Philadelphia, which I think is a pretty cool thing. 13 stories up. I think it's the highest GMP manufacturing facility that I know of. But we've been able to not only control our own destiny in that context, but we have built a unique expertise that now we have licensed at 38th and Market, the only commercial facility in the United States approved to make a gene therapy. So it's a huge endeavor and an amazing accomplishment, and it really has set us up for the long term. We've also done things that have tried to push the envelope when it comes to clinical development. I told you that mobility test is not a standard test that ophthalmologists use in their clinics. We innovated on that. We worked very closely with the regulators to do that. And we also engaged early with payers, and you see some of the fruits of that in terms of what we just announced this month. And we have been steadfast in trying to come up with new ways to ultimately diagnose and deliver and ensure access for patients, whether that's through these centers of excellence, whether that's through the patient support and assistance programs, whether that's through these innovative contracting models that we have pushed for. So let me close with that and just tell you a little bit about the way we've thought about our company, and I think this is very much a part of our success. We have a motto at the business that we say we don't follow footsteps, we create the path. It is not for the faint of heart to be in biotechnology, period. I think it's even less for the faint of heart to be in an area where no one's ever tread before, and you're constantly looking into the void. And so you have to have an attitude that you're willing to challenge the status quo, the way things have been done, and you're willing to take on that risk. But I think it is incredibly gratifying and exciting to be at a point where you're actually writing the script and writing the history of how this stuff works, and I'm certainly proud that we're doing it here in Philadelphia. So let me conclude with that, and now I'll take any questions that you have. Thanks.
Let this gentleman ask his second question before I come back to you.
I was wondering if you could throw more light on the chemistry of the enzyme, whether based on some of the observations you made, it sounded like it's not probably completely knocked out catalytically. It's only a specific activity going down. I was more interested in that, and also a word of congratulations. I made the same travel in West York and Penn.
Yep.
And when we published on gene therapy, one event at Penn
Yep.
sounded the death knell. So you should have had a lot more courage to do that.
Well, so thanks. And there's certainly there are a lot of people who worked hard for many years and persevered for many years. I mean, two people I'll note in particular who have really been the partners for me in this journey. The first is Kathy High, was my scientific co-founder in this business, is the president and head of R&D for us today, and is someone who's dedicated, really her professional career and her life to trying to address genetic diseases through various technology, including gene therapy. I mean, she deserves a tremendous amount of credit for persevering when it looked pretty dark, especially in this area of the world. The second is Steve Altschuler, who was the CEO of the Children's Hospital of Philadelphia, who not only saw Kathy's vision, was willing to fund a center dedicated to cellular and molecular therapeutics in 2004 when everyone else was running for the hills and not investing in this. And Steve deserves not only a lot of credit for investing in that, but then choosing to invest in this idea of Spark, which I'll say one thing about that. Not only did he invest in myself and in Kathy to go and create this business, but he was bold enough to convince the board of trustees of CHOP that CHOP should invest its own money into Spark. And so I'm proud to say that CHOP's $35 million that it put into Spark is worth about $500 million all up. So you do bold moves like that, and you trust people, it pays off. So those two people I think deserve a tremendous amount of credit for this. I would say about the chemistry, I think if I got your question, you're asking about RP65. So because you aren't integrating into the host cell genome, you're not editing it, right? So we don't do gene editing. We don't touch the host cell genome. We're adding a normal functional copy alongside the rest of all those chromosomes. And so therefore, yes, if a patient has some residual activity, like they don't have a null mutation, they're probably spitting out something, they're kicking off something, and so we're just adding in amount of expression on top of it. So if you think about it in the context of like hemophilia, you can measure, you can't do this in the eye without, you know, a biopsy, which we don't do, but in the case of hemophilia, those patients had about, you know, they're severe patients, so they all have less than 1% or so, 2 to 1% activity. So they're producing some of it, but not enough, and they have severe disease because they're not producing enough. And so we're then adding, you know, 30% on top of that to get them to mild or normal, and that's the concept behind it.
If I could just add a note to our streaming participants, you can tweet questions to us, and Anthony will read them out, and we have a couple more questions here on the floor.
I'm glad I don't have to tweet responses. I can just say something.
Well, first, I want to congratulate you for bringing a amazing revolution in gene therapy in this world, basically. So I have one question. So you are AAV2. Which kind of serotype for is neuronal specific? That is AAV2. Are you considering?
For the retina
Yeah.
application, it's
And what are the challenges you are facing for manufacturing? Because for human, we need a huge number of like amount of virus we need for trials, anything for treatment. So what are the challenges you are facing? Because I prepared viruses, I know the steps. So it's really hard because for transfixing, like several plates, and then you getting very limited volume of viruses, purified viruses, right? So what are the challenges for pulling it off for a big scale?
So a couple things. So one of the advantages of the eye is, and when you think of 3 compounding factors which makes manufacturing challenge completely different, especially in the eye, you talk about a one-time treatment to a rare population in a small compartment that you only need, as I said, 150 billion particles, vector genome particles in 300 microliter solution. You can actually produce enough, even in single batch runs, to more than meet your demand. And that's a function partially of one of the things we in-licensed and brought, actually assigned from CHOP, was the know-how of this manufacturing process, which was a highly efficient adherent cell culture-based system upstream and a cesium gradient-based process downstream. And that's what we've taken through the FDA. We've spent about 3.5 years not only building that facility from May to October of 2014, but then since October 2014 to August of 2017, in less than 3 years, we basically validated that process, developed more than 24 analytical methods, and basically passed the first ever pre-approval inspection. So I mean, our team did an amazing job getting us to this point. Now, in terms of some of our other applications, so we now have that one system. We're also working on another system today where we're trying to adapt the cells to exist in suspension. So rather than actually, what an adherent process means is that you actually attach the cells to the outside of what we use, or these roller bottle technology, but it essentially adheres to the outside. What we're now moving to is to try to put a completely different set of nutrient and chemistry around so the cells can actually exist and float in what's called a bioreactor, and therefore you can then scale up geometrically and go to much larger scales. And so we actually have already established that process, innovated on that, patent on that, and we're actually in the middle of the scale up. And we'll use a suspension process for things like hemophilia A, which is not only more vector genomes per patient, but is obviously more patients, right? There are about 16,000 people in the United States with hemophilia A, and depending upon how you cut it, about 100,000 people in markets that people typically would market into, and that contrasts with more like 6,000 for RP65.
So hi, it's a very nice story. It's fantastic. I have a couple of questions for you. The first one is because the way you formulated it as it's a single intervention, and with a requirement for a long-term durability of how effective it is.
Yep.
I wonder if you can comment on how that changes the way you go about your phase 3s going forward, yeah? And if that changes the way we do this therapeutic development, because now we're talking about once you give an intervention, we are waiting 3-5 years to even figure out one of those endpoints, yeah, so to speak, at least the key endpoint anyway for this kind of an approach.
Yeah.
So how does that impact the way you do business, so to speak, if your phase 3 is going to have to wait 5 years each time?
Well, so you're making a really important point, and I would distinguish between what the regulators need to approve a product versus what a payer, an insurance company, might want to see to pay for a product. Those are two different things. And so from a regulatory perspective, the FDA and other agencies are tasked with ensuring that products are safe and that they're effective. And you can establish safety and efficacy in a shorter study. So in something like factor 9 or factor 8 for hemophilia, I just showed you that in 1 year you can show a 97% reduction in infusion rates and a 99% reduction in infusion rates. That's more than a big enough effect size to power a trial and show statistical significance. What you're getting at is that you also want to build a data set to show that over the long term the product continues to maintain its effect. And so that's why, and one of the things that we learned from RP65, which is sort of further your question, the sooner you can actually use the same endpoints that you'll use for registration, even in your earliest trials, then I can answer your question and say it's the same endpoint was used in the first phase 1, and when I finally got to approval 5 years later, I can say that patient. And so for that, if you take the hemophilia example, all those 10 hemophilia B patients that were in the phase 1/2 trial, as we move to phase 3, we'll still be tracking all of them. So while our phase 3 trial may only have a year to an endpoint or whatever it might be, those patients might be 3 years, 4 years, 5 years, and you start to establish a track record. The other thing I think you can do is to try to establish risk-based agreements like what we're saying, right? So we're willing to stand behind the product that it keeps working, and so I think when you stand behind your product, you'll ultimately be able to get access and pricing for it, and that's sort of what we're trying to use all those elements together.
Jeff, do you have time for a couple
Sure,
more
yeah.
questions? It's 10 after 1. I just want to
Yeah,
make
I'm
sure.
good. Yeah.
Hi, for the CNS-related studies, I understand that you target cells post-mitotically, and the whole drinking thing, cells do die, but with synaptic plasticity, there's also new growth. So
Yep.
for the CNS treatments, do you envision that being one treatment over a lifetime or one treatment every certain number of years? Or do you think that that kind of new growth wouldn't be significant?
Well, I think it's all kind of, it depends, right? So first of all, I think that's a question that we're going to need to answer over time by following, by doing clinical investigation and by following patients. Just to give you an instance, yeah, so for something like Batten's disease, what we're doing is we're actually doing an intracerebral ventricular injection. So it's a single injections, I believe it's a single injection site. So it's a neurosurgical procedure, one-time injection of some number of viral vectors per chamber, I believe, but I don't know, we're still probably working on the dosing is my guess. But we're trying to actually transduce the ependymal cells that are lining the ventricles and use them essentially as a depot to secrete, in the case of this disease, what's called TPP1, which don't ask me to tell you what TPP stands for. It's a peptidase, but I don't remember which one it is. So you can get cross-correction in that disorder. So you can secrete it out into the CSF and then kind of use the plumbing, if you will, of the CSF to have it biodistribute throughout the brain. And so if you had new growth, as long as you still have the cells with the copies of the gene in the ependyma, and it all depends upon how much you need. So going to the hemophilia example, getting someone from 1% to 5% of normal is actually moving them from severe to moderate. Getting them, actually from severe to mild. Moderate's 2 to 5. And so in the case of TPP1, I don't know the exact percentage, but you don't actually have to get every cell to be producing every or a certain amount to change the actual pathophysiology of the disease, and that's sort of the goal ultimately. So that's why it depends, meaning in part because what disease you're picking, how much protein you have to produce, is it secreted, is it not secreted, is it an intracellular versus an extracellular protein, all those things sort of go into the calculation of what we're picking.
So Jeff, I want to ask you a question that's sort of more macro than micro, and
Yeah.
then I think there's going to be a more micro question in a moment. Could you just comment broadly on the field of genetic research? Most of your focus appears to be around orphan diseases,
Yep.
diseases that live in subpopulations. Wondering if you could comment on the big picture of diseases like Alzheimer's, which are such a big deal for the
Yeah.
American economy and for families. And we know that there is some genetic connectivity.
Yep.
It's poorly understood. There's a lot of things that we don't get. Our chairman of neurology is here. I'm sure he would have lots of comments on this. So if I could just bring it back up to
Yeah.
a macro level, and if you could just say a few words on what you think the state of the science is and what we could anticipate in the future.
Well, I think, so it goes under the theme of, let's walk before we run, which is that monogenic disorders are places where we understand a direct connection between a gene defect and biology. And if we can correct that monogenic disorder by giving a correct copy of a gene or suppressing too much production of a gene product or knocking in something, and so what I just said in those last two things are beyond what we have done, but that's sort of what some people are trying to do with gene editing. And so, but all of that, you put that under the lens of genetic medicine, they're all still focusing largely on monogenic disorders because there is a direct relationship that we understand about the disease between the gene and the biology and the pathophysiology. There's no question that at some point in the future, you will see people, I think hopefully successfully, try genetic strategies to common disorders. We've actually seen people try it. There was a company a couple years ago that tried to develop a treatment for wet age-related macular degeneration, which is not a genetic disorder, but they tried to mimic the anti-VEGF treatments that are on the market as monoclonal antibodies. We have to get repeated intravitreal injections. That didn't work. Now, there's probably a whole host of reasons it didn't work. There was another group that tried to work on heart disease and coming up with a gene therapy single-dose treatment for heart disease. It also didn't work. So I think there's some, and in the context of this sort of today's talk, how do you build a successful biotech company? I think our view is that you need to walk before you run, and so success will beget success. Those other two companies don't exist. So there's an argument about you pick your spot where you swing for the fence. I say this to the team all the time. If you bunt or single and you get on base, you're still kind of in play. If you swing for the fence and miss, you're probably not. And so there's a way to build that success, which then helps you raise more capital, $848 million or whatever we've raised, that you can then build on that, and you can let the understanding of the biology of the disease progress. And I don't think in some of these disorders that we fully understand it well enough, but I'm sure we will. And as we will, and as we've advanced these tools, then we'll be able to marry up the understanding of the biology of a common disorder and how it relates to genetics. I mean, I know, for example, in Alzheimer's disease, there's people that have published on strategies trying to use ApoE2, which is a variant that seems to be, that has been described to be protective, and there's natural history data of patients, was it from Northern Europe, that I think are more well protected who have ApoE2 mutants than the people who are ApoE3, ApoE4. So could you actually deliver ApoE2? Again, I think it's a question of, do we understand the biological relationship between the delivery of that as a gene versus just sort of understanding the genetics of risk, and that's two different things. Yeah.
Thanks for your presentation.
Welcome.
Switching gears a little bit towards the market access piece.
Yep.
So I think it's really awesome that you guys are willing to stand behind your product and
Yep.
engage in the value-based contracting.
Yep.
Could you tell us a little bit about how you guys are collecting the data and how you get those types of endpoints, since they're clinical endpoints and you're not really capturing that through medical records or lab data?
So it's a great point. And so for something like Luxturna, one of the benefits as well that we are doing this at a select number of centers is that we can, what we're doing with that is we're using the full field light sensitivity threshold testing measure, which is an instrument that most, if not all, ophthalmologists have access to. So we take a measurement at baseline, and then we take a measurement 30 to 90 days later in their normal course of follow-up, and then we take a measure 30 months later. And because we're doing it at a small number of centers, you can see how you can collect that clinical data for a small population. It certainly raises the question, how do you do that and scale it? But when you go to something like hemophilia, one of the unique things about hemophilia is you can measure the indirect effect of your gene therapy by seeing whether or not the patient needed infusions. Right? The patient's not all of a sudden going to go from being a hemophilia patient that needed 100 infusions a year to needing no infusions a year if they didn't have something that happened. And so the idea of a gene therapy intervening and being able to track the infusion rates in those patients would be a way you could actually see that without having to get the patient to come back in, without having to go to clinical records, but you could see it through claims data. So each application, I think, will have a different situation on how you do that, and that's a lot of what is being discussed. And so myself and a few other CEOs of gene therapy companies are working with Mark McClelland and Duke University on exploring what are everything from the legal to the logistical to the data. How do you actually make these value-based agreements come to play? And there's a lot of energy around that, which is good.
So this will be our last question.
So we do have a question from Twitter.
Okay.
At KanayoMD asks, how would someone with a potential investigational new drug raise a secure similar investment like you did, but without ties to CHOP?
Well, so I think the most important thing is depending upon the stage of where that investigational agent is about the data and about sort of do you have a consistent set of data that can hold together a story on why you think it will be successful. In this world today, which I think will be the case for many years, the investment community typically wants to understand for a biotech business, what's the risk that you will be able to accomplish the next stage? And largely, when you start a company, it's a question of will the next step in your clinical trial show positive results? Will it be safe and will it show efficacy? And so if you are someone who doesn't have clinical data yet, who doesn't have information yet in patients, then it's a question of how much data have you collected in your preclinical experiments, and have you answered all the potential questions that an investor might ask? So for example, if you have data in mice, that's generally going to be harder to convince somebody than if it happens to be that there's a large, naturally occurring, larger model of that disease. It turns out for RP65, this Swedish Briard dog has RP65. Right? So the commonalities and believed perception of the risk of translating from a larger animal model to a human experiment is perceived lower risk than if you're in a mouse model. Right? And so whether it's that, whether you've showed enough data to show when you're going to hit toxicity, well, any of that data package is ultimately, in the earliest stages of biotech company, the most important thing to be able to show, have you answered all those questions that an investor will ask, and do they want to take that risk investment?
Jeff, thank you so much
You're
for
welcome.
being so generous with your
Yeah.
time, for coming to Jefferson, for participating in our series, and for answering some very pointed and detailed questions.
Yeah, happy to do it.
Thanks.
Thanks for having me.
Thanks,
Appreciate it.
everybody.
Yep.